Hello, everyone. My name is Kathleen Montoya. In this poster, I present the project I am developing during my postdoctoral stay at the Autonomous University of Baja California. The project is titled THERMO and pH-Sensitive Polymeric Micelles containing salicylic acid as a prodrug, and is being carried out in collaboration with the Technological Institute of Tijuana. The main objective of this work is to develop prodrug polymeric micelles with potential use as drug nanocarriers that respond to changes in temperature and pH. Well-defined block copolymers have been synthesized using the reversible addition fragmentation chain transfer polymerization technique, known by its acronym, the RAFT technique. These copolymers feature hydrophilic blocks derived from the statistical copolymerization between diethylene glycol, methyl ether methacrylate, known as DEGMA, and oligoethylene glycol, methyl ether methacrylate, known as OEGMA, and hydrophobic blocks derived from the statistical copolymerization between butyl methacrylate and 2-methacryloyloxy benzoic acid, which we will see abbreviated as BUME and 2-MO-BZA, respectively. The hydrophilic blocks have thermosensitive characteristics, while the hydrophobic blocks respond to pH and can also release salicylic acid through a hydrolysis process. The self-assembly behavior in phosphate-buffered saline solutions was studied at different pH values and temperatures. In these studies, nanometric-sized micelles were obtained that were stable at pH less than 7.4 and showed a thermosensitive response at body temperature, which gives these copolymers the potential to load and release hydrophobic drugs. To put the project topic into context, I would like to define some concepts and provide you with a little more relevant information. In recent decades, interest in polymeric micelles has grown because they have shown great potential in preclinical models, improving the therapeutic efficacy and safety profiles of drugs. Polymeric micelles are made up of small chains of amphiphilic polymers, which can self-assemble to encapsulate hydrophobic drugs in their core and release the encapsulated drug at a specific site by destabilization of the aggregate. These can break their stability in response to an external stimulus if they are manufactured with some smart polymer. These types of polymers have gained a lot of attention due to their ability to respond to various stimuli, which gives them a wide variety of potential applications. The synthesis of smart polymers with modulable responses through the correct selection of monomers and synthesis routes is now a reality. In this context, polymers based on polyoligoethylene glycol, methyl ether methacrylates, have attracted much attention due to their biocompatibility and the fact that they present the property of modulating the value of the lower critical solution temperature and producing a tight phase transition. On the other hand, the polymerization of 2-methacryloyloxy benzoic acid is also of particular interest, as it produces a prodrug capable of undergoing hydrolysis at the label ester bond of its side chain and releasing salicylic acid in a sustained manner. Salicylic acid is recognized for its various pharmacological properties, including analgesic, anti-inflammatory, antimicrobial, and antithrombotic activities. The formation of micellar aggregates depends on several factors. However, the ratio of the length of the hydrophilic and hydrophobic block is of greater importance. RAFT polymerization offers the advantage of obtaining polymers or copolymers with narrow dispersion and achieving different complex architectures, such as blocks, stars, among others. This technique is a reversible deactivation, radical polymerization, RDRP process. Since its first report in 1988, it has become one of the most important techniques for the controlled synthesis of polymers. Therefore, in this work, block copolymers derived from the mentioned monomers were obtained for the first time by RAFT polymerization. These smart copolymers were self-assembled to form spherical nanomicelles with great potential in drug delivery applications, offering new opportunities for the development of more effective and specific therapeutic strategies. Raft synthesis of the block copolymers was carried out in two reaction steps. First, statistical copolymers were synthesized using the degma and oegma monomers in a molar ratio of 70-30, respectively. 4-cyano-4, propyl sulfonyl-thiocarbonyl, sulfonyl pentanoic acid, a trithiocarbonate type CTA, was used as a chain transfer agent. 4,4-azobus, 4-cyanovaleric acid, was used as an initiator. These reagents were dissolved in dioxane. The reaction mixture was bubbled with nitrogen and sealed to maintain the reaction in this atmosphere. The polymerization time was 2 hours. After this time, the reaction was purified, and the hydrophilic chains were extended with the hydrophobic block. For this purpose, the statistical copolymerization of BUMA and 2-MO-BZA was carried out, using the same reaction conditions as above. After the reaction, all the prepared polymers were purified by precipitation using dichloromethane and a mixture of ethyl ether petroleum ether. For the formation of micelles, the already purified copolymers were dissolved in THF and added dropwise to PBS solutions at pH 7.4. After 12 hours of stirring, they were placed inside dialysis membranes and dialyzed in PBS at pH 5 and 7.4. The stability of the aggregates was studied with changes in the pH of the medium and temperature. The successful production of the block copolymers was confirmed by hydrogen nuclear magnetic resonance analysis. In the spectrum obtained, the characteristic signals of the hydrogens present in the copolymer structure were identified. As priority signals, we can mention the chemical shifts around 13 and 7.5 ppm, which are attributed to the hydroxyl group, marked as the P signal, and to the aromatic protons J, K, and L. Also, the signals at 4.03 and 3.88 ppm, which correspond to the methylene groups next to the oxygen of the ester group in the side chain of the oermas and BUMA, are marked as C and F. The copolymers were also analyzed by gel permeation chromatography to determine the molecular weight and size distribution. Figure 2 shows the chromatograms obtained for a series of copolymers. A well-defined unimodal distribution is observed for the hydrophilic PDO block. After the extension with the hydrophobic block, in which variations were made in the molar ratio between BUMA and 2-MO-BZA, a shift to a shorter elution time was observed while maintaining the unimodal distribution. This indicates the obtaining of higher molecular weights with good work distribution, which verifies the successful chain extension. On the other hand, the formation of micellar aggregates was evaluated. Figure 3 shows an FE-SEM micrograph of the micelles obtained in PBS solution at pH 7.4. Small dark-colored streaks with an average diameter of 23 nm can be seen. Figure 4 shows the hydrodynamic diameter studies by DLS of the micellar solutions. Figure A shows the size distributions in PBS solution at pH 5, and Figure B shows the distributions at pH 7.4. We can see that the micelles were more stable in an acidic medium, since at pH 7.4, some of them lost stability due to the deprotonation of the acid groups. The micelles that were not affected at this pH were those that contained an acid percentage lower than 20%. The following table summarizes these results. Here we can see that the RAF copolymerization of the copolymers was controlled since dispersions of size less than 1.5 and an efficiency of the chain transfer agent close to unity were obtained. This indicates that the copolymerization could be controlled with the CTA used. The micellar aggregates in solution presented hydrodynamic diameters between 74 and 30 nm, which indicate adequate sizes for a drug nanocarrier system. In addition, the transition temperature of the micelles was corroborated, which depended on the molecular weight of the copolymers. Initially, the hydrophilic block showed a TCP of 42 degrees, which was reduced to 39 degrees with the extension of hydrophobic blocks less than 40% molar about the hydrophilic block. The critical micelle concentration was also determined. This concentration indicated the formation of micelles up to a minimum concentration of 1.35 micrograms per milliliter in PBS solution at pH 7.4 and room temperature. With these results, we can conclude that well-controlled block copolymers were obtained using the raft technique with up to 60% of THERMO BZA fed. Stable spherical micelles were observed at pH 5, however, micellar stability was compromised as the pH increased due to the presence of polyTHERMO BZA in the copolymer. In addition, the copolymers presented a TCP of 37 to 45 degrees C, with variation dependent on the pH of the medium. The findings of the study suggest that these nanomicelles have thermosensitive and pH-sensitive properties that make them potential nano-carrier systems for hydrophobic drugs. Finally, I would like to thank Conosite for the funding provided. Thank you.